Okay, hello students. This is Dr. Stefan, and today we have to talk of protein synthesis as our last part of biomolecules. So, what is protein synthesis? Protein synthesis is simply the process through which protein molecules are built up in the cell from genetic material. Okay, genetic material is information that is found in our DNA, as we saw. So it is important that you know that this information that is in our nucleus comes in the form of a code. This code is a sequence of nucleotide bases and it has the following properties. So the code is a triplet code, meaning that it is made up of 3 bases. It is degenerate, meaning that there are more codons than amino acids. Okay, so not every codon codes for an amino acid, or not every part of the code produces amino acid, so they are degenerate. Also, it is punctuated, meaning that there are presence of start codons like AUG and stop codons like UGE, UAA and UAG. So start codons are region where translation starts, whereas stop codons are regions where translation stops. Also, it is universal, meaning that it codes for amino acids in all organisms. So protein synthesis has 3 main stages. The first being transcription, the second being activation, and the third being translation. So RNA has a key role to play in protein synthesis because it is the medium of DNA synthesis between the nucleus and the cytoplasm. Remember, we saw the different forms of RNA. We said there was messenger RNA, transfer RNA, and ribosomal RNA. Okay, so during protein synthesis, in transcription, DNA double helix unwinds and exposes the nucleotide on a single strand. Then next, there is a complementary nucleotide from a messenger RNA pool that pairs up with those on the exposed DNA strand. So with the help of RNA polymerase, these nucleotides link up together and give rise to messenger RNA. So what happens is that initially we have DNA like this that unwinds and exposes some segments of the DNA that become free, and then we have the RNA. We have the RNA that some nucleotides that bind at some particular site on that exposed DNA strand, and from that portion now there is RNA polymerase, which is this enzyme that now gets into it and now add other nucleotides. Okay, so it increases the nucleotide, and at the end you have RNA that is formed, this messenger RNA that is being formed from the DNA template, from the DNA template. Okay, so after the synthesis of the messenger RNA, this messenger RNA now has to be processed and leave the nucleus for the cytoplasm. So in activation, the messenger RNA moves out of the nucleus into the cytoplasm with the help of ATP. The transfer RNA molecule is bound to amino acids, giving rise to amino acyl transfer RNA. So we have a diagram here that demonstrates to us how an amino acid is being loaded on a transfer RNA molecule. So there is this enzyme that we call amino acyl transfer RNA synthetase, which is responsible for catalyzing this reaction. So what this amino acid does is that with the help of ATP, energy from ATP, it binds to some specific amino acid, or it uses some specific amino acid as substrate. Okay, then it also it has 3 sites: the tRNA site, the ATP site and the amino acid site. So the tRNA site is where the transfer RNA itself is going to bind. The ATP site is where the ATP will bind, okay, to release energy, and the amino acid site is where the specific amino acid to the transfer RNA will bind. So during activation, okay, you have ATP molecule and the specific amino acid that binds to the enzyme. So you can observe from here, this is our ATP with our nucleoside and our 3 phosphate groups, and then 2 of these phosphate groups are being released and energy is being used, and then that energy that is being used now is used to combine to fix some specific transfer RNA, and that specific transfer RNA, some of that, when AMP, that is adenosine monophosphate, leaves, that energy is being used to bind this a specific amino acid to the specific transfer RNA to form the amino acid transfer RNA. Okay, this is the amino acid transfer RNA here that is now being released, and that can enter into translation. So the next step after the synthesis of messenger RNA, we have the messenger RNA that leaves the nucleus through the nuclear pores to enter the cytoplasm. Then in translation, the messenger RNA binds to ribosome to become a polysome or a polyribosome. Okay, so you have a messenger RNA and you have many ribosomes that bind to it, and the resultant structure is called a polysome or polyribosome. So the first messenger RNA codon initiates the chain formation by attracting an amino acid tRNA molecule. The second codon equally attracts an amino acid tRNA molecule, showing the complementary anticodon. Then the ribosome holds all the RNA in place for enzymes to link up adjacent amino acid through peptide bonds. So once an amino acid has been added to the chain, the ribosome moves one codon along the messenger RNA. The tRNA molecule leaves the cell. Reading continues until an end codon is encountered. So we gave the different end codons. So this is what happens. You have the messenger RNA that binds to a ribosome, okay. So this specific codon, they attracts some complementary codons, okay. This complementary codon that are already bound to some specific amino acid. So this the next codon also attracts another specific tRNA that contains a specific amino acid to the codon itself, and the ribosomes helps to fix them in place so that the enzyme can bind them together, and at the end you have the empty tRNA that leaves the ribosome, and you have the amino acid that is being added onto the peptide chain, the peptide chain or the polypeptide chain. So the third step now is the synthesis of the protein. So you can observe that you have different amino acids. So this amino acid, they bind to tRNA. You have the ribosome that kind of reads the messenger RNA. So as the name says, messenger, they transmit some message. So the ribosome is the one that translates that message into usable form, and the amino acid itself is being added through with the use of amino acid transfer RNA, okay. And you have the peptide, the polypeptide chain that increases correspondingly as the ribosome is continuing to read along the messenger RNA. So this is another diagrammatic representation where you have the transfer RNA bound to some amino acid, okay. So these are some other images. So for example, if we take this as our substrate, okay, remember we said DNA is always anti-parallel. meaning that one strand always leaves from 3' to 5', and the other strand leaves from 5' to 3'. And we said that the nitrogenous bases are complementary. A goes with thymine, thymine goes with adenine, cytosine goes with guanine, guanine goes with cytosine, and so on. So these are our strands. So in this region where there is mismatch, there is what? There is mutation. So there can exist some region where you will observe some mismatch where adenine can be bound to cytosine. Okay, this could be an error in replication. So this, considering that this is the original strand, the complementary strand moves in the anti-parallel direction. Okay, so the original strand is from 5' to 3'. The complementary strand is in the anti-parallel direction. Okay, so you can observe the nitrogenous pair, a pyrimidine. So these are pyrimidines here, and this is a purine here. So these pyrimidines, or these nitrogenous bases, they are bound to the sugar backbone. Okay, and this sugar backbone on its turn is bound to a phosphate group, and this here forms what we call a nucleotide, and in between these two nucleotides there is this bond here that we call phosphodiester bond.